हाँ सर, स्लाइड मोड कर लीजिए सर प्लीज इसको।
हाँ
हाँ सर।
So is it visible now?
हाँ सर, हाँ सर।
Okay, thank you so much. So first of all, I would like to thank Meadow Agriculture for giving me the opportunity to present a invited talk on the topic nanoparticle-mediated advances in improving sperm quality, livestock sperm quality, and its implication for sustainable reproduction. Okay, so first of all, let us know why I have chosen this topic. So we all know that in the present scenario, most of the insemination techniques are done through artificial insemination in livestock, right? So what we see is it is one of the most powerful and cost-effective tool in delivering or salvaging the genetics of livestock and in different animal species such as cattle, goat, small ruminants, even in swine. This technique has been used widely. Now there are several importance or benefits of artificial insemination in the current scenario. So we have it. It can have you can see there is during artificial insemination there is rapid genetic gain, and it also lowers the generation interval. Plus there is enhancement in lactation and growth since we are introducing superior quality of semen into the female. So we can see so many genetics can be changed, and it has also a global export market where semen and embryo export, it crosses more than 300 million doses per year. But the main bottleneck in this procedure is the freeze thaw damage. So many studies have shown the spermatozoa will incur around 30 to 50% of damages during the process of cryopreservation due to the presence of mostly PUFA. What is PUFA? PUFA is polyunsaturated fatty acid. So what happens during the process of freeze thaw when the temperature is lower than 5 degrees Celsius, the spermatozoa undergoes cold shock. And during this process, PUFA gets reorganized on the sperm cell membrane and it gets oxidized. And during this process, reactive oxygen species is generated. And this causes direct sperm membrane damage due to production of malondialdehyde, which is a byproduct of lipid peroxidation cascade. Then it also causes mitochondrial dysfunction, resulting in lowered ATP production and subsequent DNA strand or DNA helical damages. Now, in the pig or swine, we have seen most of the artificial insemination is done by liquid semen, not cryopreserved semen. Around 90% of the inseminations are done by liquid semen. So during this process, why in pig we do liquid semen insemination is all because of the fact that these pig semen are or the swine semen are less cryotolerant, and all these changes will be causing oxidative stress. So whenever there is cold shock in the spermatozoa during the preservation process, oxidative stress is induced, and this is the main principal bottleneck which causes sperm damages. Coming to why spermatozoa is vulnerable to such damages. First thing is the high PUFA. So it is very sensitive to the process of liquid peroxidation. Then we have the limited cytoplasmic component which has very less antioxidant reserves. And the dense mitochondrial sheet which is in the midpiece of the spermatozoa, it is a prime source of ROS. And these all cascade of reactions make the spermatozoa highly susceptible to cryogenic stress. Now, what happens during these cryo-induced damages to the spermatozoa? First thing, the reactive oxygen species will damage the PUFA-rich membrane, leading to direct decrease in sperm motility, and in turn, it affects the fertility rate. And whenever it affects the mitochondria, you will see there is membrane depolarization and decreased ATP production. Subsequently, it also damages the DNA by causing DNA strand breakage. Similarly, it also affects the acrosomal membrane and it disrupts the acrosome integrity, and it prevents premature enzyme release from the acrosome. Now, we have seen in semen extenders, we usually go for broad spectrum antibiotics. So the semen extenders are, in the semen extenders, we routinely use antibiotics such as penicillin, streptomycin, and this can cause spillover of antibiotics in the system or in the production system. So what happens is that many antibiotic resistant bacteria will be produced, and this MRSA, E. coli, these things are mostly antibiotic. These are AMR, they have significant AMR concerns. Okay, and in this way, nanoparticles, we have seen nanoparticles can be used as a dual function. So it can have both antimicrobial and antioxidant function. Examples such as silver nanoparticle, zinc nanoparticles, chitosan nanoparticles, which we will be discussing in my subsequent slides. Now, why we use nanoparticles for preserving the sperm quality? We have many different antioxidants. We call it conventional antioxidants like vitamin E, glutathione, CMC. So many things are there. But why nanoparticles? So the main rationale behind this is its high surface area. Nanoparticles, they are usually less than 100 nanometers. So in the process, they have a very high surface area so that they can readily neutralize the effects of, we can say it, superoxide radical. And it can also mimic the activities of different antioxidants, and it can also have high bioavailability in the system, such as selenium and zinc. Then it can also directly affect the spermatozoa by interacting with the sperm surface membrane, and this way it can stabilize the spermatozoa during the process of cold shock. Now, different nanoparticles have been used in the reproductive biology to augment the semen quality, such as metallic nanoparticles such as selenium, zinc oxide, gold, silver, and there are different metal oxide nanoenzymes also which have been used. Then chitosan, alginate nanoparticles which are polymeric. Then recent focus is on green synthesis of nanomaterials through plant extracts. So we'll come into that in my next slide. We'll have a few examples of nanoparticles and few examples how it has preserved sperm quality in different species. So first, selenium nanoparticles. We know that selenium is a very important component of the GPX antioxidant defense system, and selenium nanoparticles can cause reduced reactive oxygen species generation and thereby reducing the LPO. We call it repeat peroxidation. And in boar semen, these selenium nanoparticles have been used at the concentration of 1 microgram per ml, and they have found increased motility around 10 to 12% and even fertility rate after 72 hours of liquid storage. Coming to zinc oxide nanoparticles, similar to selenium nanoparticle, zinc oxide nanoparticles also have membrane stabilization effect, and they can also reduce the different apoptotic gene expression profile in the spermatozoa. And in buffalo, this thing has been studied extensively, and they have found that this supplementation of zinc oxide nanoparticles improves the post-thaw sperm quality, and it also improves the in vivo fertility rate. Now, different formulations have come up, such as humic zinc nanoformulations, where humic acid is incorporated with zinc or coated with zinc to enhance its bioavailability, and it can act against oxidants, and it can improve mitochondrial activity. But while using nanoparticles, one concern is the dose. So we have seen many studies, they have documented that nanoparticles are having both time and dose dependent effect. So the dose criteria should be taken into account before formulating it for reproduction related studies. Silver nanoparticles have also been synthesized through green synthesis technologies, and it has been used in bovine semen, and they have found good sperm quality. Then it has also got antibiotic sparing potential. So in few studies, silver nanoparticles have been used as a substitute of the conventional antibiotics in semen extenders, because These things, if it is synthesized as a green procedure, it can contribute to antimicrobial resistance mitigation, and it can also result in sustainability of the system. Gold nanoparticles have also been used as a mitochondrial membrane stabilization effect. Then it can also have ROS modulation, ROS reactive oxygen species modulation. But this thing has not been widely studied, so we have very less data on fertility yet in different livestock species. Different nanoenzymes which can mimic different antioxidants in the system has also been used, such as metal oxide nanoparticles. Metal oxide nanoparticles such as zinc oxide can mimic catalase activity. It can cerium oxide nanoparticles can mimic peroxidase activity. Euporium oxide nanoparticles can mimic superoxide dismutase activity. But still, we see there is less data available on large scale fertility trials of all these nanoparticles. Polymeric and biopolymer nanoparticles are also been designed and used recently in laboratory animals, and they have found that alginate and silver nanoparticle composite, it protected the spermatozoa during chill condition. Now this is one of the prime focus these days, where green synthesis nanoparticles and biogenic capping materials are being used to make the system or the system more environmental friendly. And we have seen many different plant extracts have been used for synthesis of different kinds of nanomaterial. Like for synthesis of zinc oxide nanoparticles, even neem extract, Aja directa indica extract can be used. Then even turmeric extract has been used for synthesis of selenium nanoparticles. And it has been seen if nanoparticles are synthesized through the green synthesis process, the cytotoxicity is lower. Now, beyond the lab, we have very few studies regarding the conception rate improvement with different kinds of nanoparticle supplementation in the extender. Though there are enough data on in vitro studies, like increase in motility by 10 to 25% after post-thaw, then decrease in lipid peroxidation indices, and increase in mitochondrial membrane potential. But there are very few studies that show us that nanoparticle supplementation can increase the conception rate after post-thaw procedures. So the main problem is the translation to the field level. It is a major gap in this scenario. Now, before translating everything to the field level, we should have a standardized semen evaluation protocol, so that the residues are not passed on to the environment and to the offspring. So basically the main things are to be done are sperm motility assessment through CASA, viability and membrane integrity through different fluorescent dyes or through flow cytometry, acrosome integrity also through flow cytometry, then ROS quantification methods. DNA fragmentation is also a very important parameter which needs to be studied, and the AI conception rate and the passover, or if any horizontal or vertical transmission is happening to the progeny, these things also needs to be studied. Now, how this nanoparticle cause toxicity? So we have seen, and in my previous slides I have told, nanoparticles show dose and time dependent toxicity. So there should be a minimum threshold of nanoparticle uses in the semen extender before it can be translated to the field level. And how this nanoparticle can cause dose dependent toxicity is, we have seen, suppose we take the example of metallic nanoparticles such as zinc oxide. So zinc oxide will supply, at higher doses, this zinc oxide will supply more zinc ions to the biological system, and in the process it will generate more of pentom reaction, and in the process OH negative ions will be generated, which will have apoptotic like events on the spermatozoa, therefore damaging the sperm quality, even DNA strand breakage and plasma membrane disintegration. So what we see is dose is a very crucial factor in determining the appropriate uses of nanomaterials in reproductive biotechnology. Even species specific considerations should be taken into account we have seen in boar spermatozoa, they are highly sensitive to cold shock. But as against bull sperm, they are more cryotolerant. Ram and rooster sperm, they exhibit different membrane lipid profiles. So we see if excess ROS is generated, there is excess of phantom-like reaction, and this disrupts the mitochondrial electron transport chain, thereby causing depletion in antioxidant enzyme levels, and even DNA damage. DNA damage will see.
Hello.
Hello,
हाँ.
hello.
कहाँ बना आपका? अच्छा.
Any problem, sir?
No, no, sir. Continue, please.
Okay. So species-specific dose validation is very much mandatory before translating these things into the field level or into any biological system. So there is a wholesome approach where oxidative stress, DNA integrity parameters, mitochondrial parameters, and apoptotic genes or maybe apoptotic pathways should be profiled before suggesting a dose. Okay, this dose is more effective at this rate. And the methodological best practices for nanoparticle uses for translation into the field level will include is characterization. So first, if possible, nanoparticles should be characterized, and it should be done with the green synthesis protocol. Size, zeta potential, and its coating should be studied. Zeta potential, those things is readily available, readily studied these days, and its stability should also be studied in the extender. For how long it is stable, and is it causing any environmental spillover? Dose and response curve should also be studied, and more importantly, the dose and time dependent effect should be characterized nicely for any nanoparticle to be used and propagated further. And fertility trials are also important equally, and if it has any effect on the embryo or any in vivo alteration in the fertility rate should also be properly documented. Now, as we know that nanoparticles are a concern at higher doses because some studies they have documented that it is toxic to the offspring, and how these things are happening, that nanoparticle can carry over its effect during the process of fertilization, and it can interact with the zona pellucida layer, causing its early cleavage, and it has direct effect on the blastocyst and the subsequent stages of implantation in the uterus. So all this happens because the nurturing embryo will be having very limited antioxidant defenses. And there are also epigenetic concerns related with the uses of nanoparticles in a biological system, since it causes DNA oxidation and DNA methylation patterns are also changed, which can have direct effect on either filial first generation or filial second generation fitness, reproductive fitness of the offspring. And there is also limited multi-generational livestock data in this regard. And how these nanoparticles are released in the environment, I can give one example where humans can have a crucial role in spreading the nanoparticles to the livestock. Suppose a livestock keeper is using a gold ornament or a silver ornament, so there is a possibility that this thing, from there the nanoparticle can get exposed to the livestock. So there is a safety concern and the environmental issue in this. And it has also got residue and food safety because metal ion residues are need to be quantified regularly, and there is no standardized residue threshold regulation in our Indian condition. And the key message in this slide which I want to portray is nanoparticle reproductive safety must be evaluated across different biological systems. So starting from the sperm to the embryo, then to the offspring, and even to the environment if any spillover is happening. Now, these things can be integrated in a precise way so that reproductive efficiency can be increased. these days there are so many methods where sperm selection, superior sperm selection is done, such as through microfluidic sperm selection techniques, then nanoparticle enhanced extenders. And there are ROS nanosensors which can detect the oxidative stress level, and even there are AI systems that can detect the appropriate dosing for any type of nanoparticle. And these are the conceptual slides where we see how this nanoparticle uses and its substitution as antibiotic in the extender can have an one health impact. So in different AI programs, nanoparticles have been used as antibiotic sparing tool. So in these traditional semen extenders, they mostly rely on broad spectrum antibiotics, then their routine prophylactic inclusion, and the high dissemination potential in the environment. But this nanoparticle enhanced system may, after incorporation into the extender, they may reduce bacterial load. They also may require very less concentration because of their higher surface charge. They can also mitigate the problem of AMR, and it can also reduce the antimicrobial residues that are directly released into the farm. So all these things can together generate a one health perspective which takes into account the animal health, environmental health, and the public health. So responsible nanoparticle adoption may contribute to integrated One Health sustainability framework. Now, this nanoparticle uses can also have an effect on the carbon footprint. So this is a conceptual slide where how I wanted to show how improved fertility outcome after nanoparticle uses can translate to climate gains. So improved semen resilience with the use of nanoparticle and increased conception rate will directly reduce repeated breeding cycles in livestock. Then it will extend the calving interval, then synchronization intervals, and it will reduce the farm resource input. And regarding the sustainability implication, since the amount of methane is quantified through the total methane generation by total livestock input. So in this way, we can see if we have improved conception rate after uses of nanoparticles, we'll see directly there is fewer inseminations per pregnancy, and indirectly it will reduce the denominator, that is the total input per kg total livestock input per kg of milk or meat. So directly it is reducing the GHG intensity. So which is of a particular concern these days, since our government is focusing on cleaner India and greener India. So, and these all outcomes are aligned with FAO sustainable intensification goals. Now, I wanted to show how this artificial insemination efficiency can be optimized. Okay, so in the sustainability perspective, we can see improved semen preservation protocols will reduce the amount of liquid nitrogen, transport related emissions. Then it will also lower repeated AI procedures, which is the main concern these days, because it is widespread in the field condition where people are inseminating the cow or the animal twice. Then it will also reduce hormonal treatment, and at the system level, semen quality will be very good. Then there will be fewer failed insemination and improved productivity per unit of resource invested. So the main message in this slide is reproductive nanotechnology should not only improve fertility, but it must be embedded in a sustainable production ecosystem so that there is less environmental spillover, and it can have a system level impact by lowering the by improving the productivity per unit of resource invested. Now, in the circular bioeconomy framework, how nanoparticles fit into the system, we have seen the green synthesis using plant extract is already a sustainable and eco-friendly procedure. Even biogenetic synthesis have been tried from agricultural bioproducts. I have seen one paper where they have synthesized nanoparticle, especially metallic and silver nanoparticles, through rice husk, which are generated from the agricultural system. And All these things will directly result in sustainability of the system, and its impact when we see it will have a lower chemical synthesis footprint in the ecosystem. Then hazardous byproducts are also reduced, and it will align with our bioeconomy policy goals. So reproductive nanotechnology should not only improve fertility in the long run, but it should always have a ecosystem which through which it will be sustainable. Okay, so now the main thing, I have skipped few slides because those are not relevant. Now coming to the research gap. So overall, whatever studies are there, the main research gap is large scale fertility trials are have not been addressed in those because of the toxicity concern. Even the offspring safety data is missing. Ecotoxicological studies such as environmental spillover from the agricultural system, then from human to animal, how this nanoparticle can cause surface exposure, those things are missing. Then standardized nanoparticle reporting, especially the time and dose dependent cytotoxic effects are have not been documented. Then the regulatory pathway, we don't have a standardized regulation which says that this much nanoparticle usage should be done and this much should be spill over to the environment. Now, on the conclusion slides, I would like to say these nanoparticles, no doubt, nanobiotechnology is having one of the most promising future in today's reproductive biotechnology. Through, but the thing is, the toxicity related and the dose things should be standardized before translating it into the field. So, in one line, I would like to say their nanotechnology is very promising in the lab metrics, but at the field level, there should be some regulatory policies that will determine its what we say, like it is not hazardous to the environment. That should determine that it is not hazardous to the environment and it does not leave any spillover in the environment. So since my talk was on nanotechnology, we have published few studies. I have told in my previous slide, nanoparticles can interact with the sperm surface membrane. So in the first picture, you can see I have tried zinc oxide nanoparticles, and that is the plasma membrane where we see that nanoparticles may not get internalized in the system, but may act through various different ways by stabilizing the membrane, but it might not be able to penetrate the membrane. In the second picture, you can see agglomerates of selenium nanoparticles. It was during which was it was in a picture where we have induced post-thaw damages to the spermatozoa, and we have seen selenium nanoparticles can internalize into the system, or you can say selenium nanoparticles can exert its antioxidant effects by internalizing into the system. Okay, so thank you. Any questions? Hello. The session is open for questions, so if anyone has any query, I can address it now.